Hello, my name is Aaron Nupper, so I'll be presenting the research project I did for ecology, where I studied the productivity of oyster mushrooms that are grown on different substrates. So let's begin. Fungi play an important role in ecosystems. They're responsible for decomposing about 90% of plant material, which helps recycle nutrients back into the soil. They're also useful to humans, not just as food, but in medicine. For example, penicillin comes from fungi. And oyster mushrooms themselves have been shown to have potential health benefits as well. Because of this, mushrooms have become more important in agriculture, especially since they grow quickly and can be cultivated on waste materials. So in my experiment, I wanted to answer a simple question, which substrate would be best for increasing the productivity of the oyster mushrooms. I focused on the golden oyster mushroom, which is relatively easy to grow and well suited for Florida's climate. The four substrates that I tested were coffee grounds, aspen wood shavings, cardboard, and straw. Straw was used as my control because I had done some research and found that that was generally the consensus. The consensus was that that was the ideal substrate for golden oyster mushrooms. And so my hypothesis was that the aspen wood shavings would produce the fastest and most productive growth because they contain both cellulose and lignin, which fungi are very good at breaking down. So how did I do this? So to test this, I set up mushroom grow bags. So they were just big plastic bags that allowed air to flow in and out. And I filled each bag with about 1,000 grams of my substrate plus 300 grams of grain spawn, which is just grain that's already got the mycelium of the golden oyster mushrooms growing on it. So put those in the bag. And well, before I put the grain spawn in, I first sterilized the substrate. So I took boiling water, put it into the substrate, not in the bag, but in the substrate to kill any unwanted microorganisms to try and make sure it is sterile. And then I would take the substrate, put it in the grow bag, take the grain spawn, put that in the grow bag, and then seal it off and put it in a warm, dark environment to allow the mycelium to grow. Once the bags were fully colonized, I cut openings into them and moved them outside into a shaded area, which triggered the mushroom growth. So once mushrooms began growing, I measured productivity in 3 ways. I counted the number of mushroom clusters, counted the height of the clusters, and then I counted the total number of the mushrooms themselves. This process was repeated for 4 trials to improve reliability. So looking at the results, there was a clear difference between the substrates during both the incubation phase, which that's when just the mycelium was growing on the substrate, and the mushroom growth phase. So after mycelium had completely filled the grow bag, then the mushroom started growing after that. So that's the mushroom growth phase. During the incubation phase, I tracked how much mass each grow bag lost over time to basically keep track of the decomposition that was occurring with the mycelium. Aspen wood shavings, cardboard, and straw all showed similar trends, each losing about 28 grams over roughly 18 days. This suggests healthy and consistent mycelium growth. In contrast, coffee grounds lost about half as much mass and showed very little change for the first 10 to 11 days. After that, there was a sudden drop in mass, but this was because there was actually mold contamination in the bag, and not because the actual golden oyster mushroom mycelium was growing. This indicates that coffee grounds was not a suitable substrate. In the mushroom growth phase, the differences became even more clear. Cardboard produced the best overall results. It reached maturity the fastest, around 9 days in one trial, compared to 14 days for straw and 15 days for aspen shavings. Mushroom height, all successful substrates were similar, with clusters reaching about 14 to 15 centimeters tall, averaging around 6 to 7 centimeters. Biggest difference was in the total number of mushrooms. Cardboard produced nearly twice as many mushrooms as straw and aspen, which both averaged around 60 mushrooms, while coffee grounds produced none. There was also some fluctuations. For example, straw briefly reached about 100 mushrooms in one trial before experiencing a die-off and regrowth. This was due to a cold snap that happened during the trial. Overall, statistical analysis confirmed that substrate type had a significant effect on all measured outcomes, and the data clearly shows that cardboard was the most productive substrate, while coffee grounds was the least effective. So these results show that coffee grounds are not a good substrate for growing oyster mushrooms, likely because they are more prone to contamination by mold. More importantly, cardboard turned out to be the most productive substrate, which was unexpected. This actually contradicts my hypothesis, since I predicted aspen wood shavings would perform best. It also differs from some previous studies where they had found that a more wood-based substrate, like sawdust, was optimal. One possible explanation is that cardboard provides an ideal balance of structure, moisture retention, and accessible cellulose to the fungi. Environmental factors like temperature fluctuations may have also affected growth. Like I said, that was a cold snap at one point during the trials, especially during cold periods when some mushroom clusters died off. So in conclusion, the experiment shows that substrate type significantly affects mushroom growth. Coffee grounds are ineffective for this purpose, and cardboard is the most productive substrate among these tested. These findings suggest this low-cost recyclable materials like cardboard could be used effectively for mushroom cultivation. If oyster mushroom farming were expanded commercially, I would recommend using cardboard in a controlled environment with stable temperatures and indirect light. Overall, this research helps contribute to more efficient and sustainable methods of mushroom production.